Welcome all to the Faculty Research Days. My name is Wouter Maes and I'm professor at the Department of Plants and Crops and also co-heading the UAEV Research Center at Ghent University. And I will try to convince you today that plant sensing makes sense for sustainable agriculture. And we are going to cover 3 scale levels of where we are applying plant sensing in our department. The first scale level is at individual plants, where we can monitor these individual plants with continuous sensors. One of such sensors is a sap flow sensor, measuring transpiration, and the other sensor that we will talk about is measuring the diameter or the stem growth, but in such a fine detail that we can estimate daily shrinkage and daily growth. Another scale level is where we are looking at individual plants, but at very high detail with remote sensing cameras, so we can get the reflectance of these different plants. The third scale level handles on field scale, where we will be applying drones to map entire fields and get information at the field level. But let's start with the plant level first. So this is research from my colleague Kati Steppe. And we will just start with explaining how these sensors can be used for estimating drought. This is an apple tree, and we have put such a stem diameter measurement on a stem, and this is the normal pattern that you could see. So you can see that for a normal control plant without any drought stress, there's a daily growth, but sometimes during the day there's also a shrinkage if the water consumption is very high. Now, what happens if we add a plant that we put under stress? So we stop irrigation at a certain time. So what happens to the stem diameter growth? This is in red. What we can see is during the first days there's still growth, although less than a control plant, but after a few days there's a lot more shrinkage, daily shrinkage, and after a while the growth stops and actually the shrinkage, the plant starts shrinking in net. And when we see this, we can't see anything else with on the leaf level. So this is a very early signal of drought stress. So this is where we can use this stem diameter sensor. Another example, here we have made empirical model based on this sensor. This is on Ficus benjamina, and we have made a model that gives us the expected stem growth. This is for 1 day. We can apply this for many days, and we can see that on some occasions we see that there is stress, and then in the greenhouse can be adjusted so that the plants either get water or the air becomes more humid. We can also take a next step where we are going to apply this on a mechanistical model, so we have a better understanding of what's actually happening in the stems. So again, we're going to use sap flow and stem diameter measurements. We're going to feed this into a plant mechanistic model to understand it, and based on this expected water potential that we are estimating, we can then perform plant-based irrigation, not just the timing but also the quantity of irrigation that is required. So these sensors are not only useful for detecting drought stress, we can also apply them for different applications. This is an example where we are detecting root pressure detection. This is very relevant, for instance, in tomato. In conditions where there's root pressure, the tomato actually gets damaged and is economically lost. So with this mechanistic model, we can actually see when the root pressure is taking place, and this time the greenhouse conditions can be altered so to prevent this damage. Okay, let's move on to the next level where we are going to look at the individual plants with remote sensing, and this is the research by the group of LAMP from my colleague Kris Audenaert, here presented in a video.
Since the dawn of agriculture, agronomists have aimed to improve the yield and health of crops by introducing novel traits or improving existing ones. To this end, phenomics has become an indispensable tool for both breeders and farmers in the field. At LAMP, we perform state-of-the-art phenotyping by use of the Pathoviewer, which combines automated high-resolution multispectral imaging with customized data gathering. These are used to identify biochemically relevant measurements from plant images. This allows us to reach beyond the mere visualization of phenomena and obtain quantitative data. The Pathoviewer is a phenotyping platform housed in a highly controllable growth chamber, specifically developed to study the interactions of plants within their natural or unnatural extreme environments. Outfitted with high-power LEDs and precision nozzles, the Pathoviewer can meet highly specific demands for a range of experiments. Using high-resolution spectral imaging comprising of different wavelengths, the Pathoviewer can visualize the impact of biotic and abiotic stresses. Additionally, the system can measure the impact of biostimulants or biocontrol agents on a large variety of plants. Through a large interdisciplinary network at Ghent University, numerous plant pathogen model systems are studied. Thanks to the automated sensor-to-plant concept, the dynamic interaction of plants with pathogens, abiotic stresses, or biostimulants can be visualized throughout the plant in time-lapse experiments. Additionally, at LAMP, we combine phenomics with genome editing techniques, which allows us to modify or tag plant pathogens and beneficial microbes. This enables us to investigate plant-microbe interactions with a higher degree of resolution. The Pathoviewer enables high-throughput screenings for new bioactive molecules or new agrochemicals with herbicidal or biostimulatory effects. This is accomplished using bioreporter plants such as Lemna minor and Arabidopsis thaliana. LAMP consists of a team of researchers with expertise on image processing who can develop customer-tailored software to visualize or quantify specific traits. Pathoviewer provides scientifically sound insight into the physiology of plants and their continuously changing agro-ecosystems. This system provides a necessary platform for the safeguarding and improvement of globally relevant agronomically beneficial traits.
In the third level, we are going to use drones for remote sensing at field scale. So why are drones useful in agriculture? Well, first of all, in the context of integrated pest management, my colleague Peter Spanhoger will immediately tell us more about this. The three key elements are prevention, monitoring and intervention of a pest and disease. And particularly in the monitoring stage, drones can be very useful and needed. Second, in precision agriculture, we want to have a spatially explicit management. So we need to know the spatial variation of nutrient levels, abiotic or biotic stress. And this is where drones can also be useful. So in this context, we want to convert this sort of a map, the map that we create with a drone, to a task map that a farmer can then use. So what tools do we have available for our drone remote sensing? First of all, we can use high-resolution RGB or commercial normal cameras, and we can put them on drones. So one example here, we have taken a very high-resolution image of a cornfield in early June, so the corn is just emerging, and we want to map how much weeds there are in the field. So with some deep learning automated process, we can first separate the ground from the plants, then identify the rows, create a bond, and then identify all the weeds that are present in the field. This is research from my colleague, or supervised by my colleague Jan Pieters. Another example, here we have not been using an RGB camera, but we have changed the filter of the red so that it becomes sensitive to the near infrared. And this is an example of over potato. And if you zoom in, we can have a deep learning algorithm that detects these spots on the leaves. And these spots are actually indicative of Alternaria infection. So we can use high resolution RGB data for weed detection, but also for detecting diseases and pests, and for other applications such as fruit and flower count. Another tool that we have available is the 3D vegetation model. Now we can use LiDAR for this, but LiDAR is quite expensive. So we actually don't need LiDAR because we can also get a 3D vegetation model from the overlapping imagery which we obtain with a drone. And just to illustrate, this is an example of a canyon that we have been mapping with an RGB camera on a drone in Australia, a very inaccessible canyon, but you can see the 3D structure of the vegetation and the canyon popping out really clearly. So this just shows how powerful the 3D reconstruction is that we can perform. And just to give an example of this, this is a grapevine field. With just the RGB imagery, we can also obtain a highly detailed digital terrain model. How can we apply this? Well, for instance, this was a potato crop where there was some fungal infection. You can see these open spots in the field. But if you look at the 3D terrain model, so at the vegetation height, you can see that there are areas where there's much lower growth. So the plants have remained much lower due to the disease, and actually these areas are much larger than just the open spots that we could see. So the 3D vegetation model from the RGB cameras, we can use this for canopy biomass, canopy structure, and extent and severity of stress. Another tool that we have available is working towards reflectance, and we can get reflectance from either multispectral cameras on the left or hyperspectral on the right. Now with these reflectance or with these multispectral cameras, we can get a typically well-known vegetation indices. NDVI is one of these examples that is well known, and we can relate this to crop properties. With hyperspectral cameras, we can go one step further, and we can see this is the expected reflectance for a normal healthy canopy, but we can then detect this kind of different spectra, and this we can relate very well to a disease, conditions of abiotic or biotic stress, or a limitation of nutrients. So these reflectance products we can use for canopy condition, for assessing nutrient and micronutrient contents, but also for disease and weed detection. A final tool that we have available is thermal imaging. Why can we use thermal cameras for this application? Because this is showing that the transpiration on the x-axis is actually linearly related to the canopy temperature on the y-axis. So the higher the temperature is in given conditions, the less the crop is transpiring. So one illustration of this is again the same grapevine field, and we can see that even for such a small area, we can see that there's quite big differences in surface temperature, which means that the area on the left here is under severe drought stress, whereas the area on the right is still very healthy and transpiring at full rate. So we can apply thermal imaging for drought stress. It's not limited to drought stress. This is an example of a kiwi fruit, very high detail. We can see that there are some brown spots on the leaves, but if we look at the thermal camera, we can see much more spots popping up, and these are indicative of, in this case, a bacterial infection. So thermal imaging we can use for drought detection, but also for disease detection. So if we sum up everything, we can use drones with different kind of sensor for a lot of very relevant applications of precision agriculture or IPM applications for early weed detection, nutrient status, pathogen and pest detection, drought stress detection, and for other applications as well. Okay, then we end with a short word on our UAEV research center, which I am co-leading. So far we have been talking about the applied research, but in this research center we are teaming up with people from the civil engineering who add their technology knowledge. And this gives new possibilities for our research. For instance, this is research in which we are improving the state-of-the-art for the thermal image processing. On the left, this is the normal procedure with a lot of flaws in it, and on the right, the improved procedure. Another example, here we are investigating how we can still measure multispectral or hyperspectral data if conditions are not constant, which means if solar illumination is not constant. This is shown here, for instance, in the darker spots over the field. Now we can correct for that. This is the same image, but then with corrections, so we can remove these influences of solar irradiation. Other examples include, for instance, sensor fusion. So we can have different sensors and overlap them completely, for instance, thermal and hyperspectral, or perform sharpening, or we are also responsible of performing the hyperspectral camera processing pipeline of a commercial hyperspectral camera. So with this combination of our applied and technological level, we can do well, state-of-the-art research. And there's one more layer to it. We're not only combining our knowledge, but we're also providing a service. So we are performing flights and we are putting our state-of-the-art infrastructure available for anybody who's interested in this. Okay, this was the end of the presentation. I hope you have questions for us, so please go ahead and ask them.